Our next guest is Matt Lazarus, VP of Business Development of Pasithea. I'm very excited to have Matt here. I've known Matt for now a pretty long time. He is a Tulane graduate as well, so he's very familiar with these streets. Pasithea is developing PAS-004, which is a next-generation oral MEK inhibitor with a unique macrocyclic design. Design. It offers a much longer half-life and a more stable drug levels than existing MEK inhibitors, which could translate into better efficacy with fewer side effects. Right now, PAS-004 is in a phase 1 trial in patients with advanced solid tumors driven by the MAPK pathway mutations, including RAS, RAF, and NF1. Early data shows a clean safety profile with no dose-limiting toxicities, rash, or ocular side effects. One patient with BRAF mutant colon cancer, typically unresponsive to MEK monotherapy, has remained stable for over 6 months. The company expects to report interim clinical data this year, which could be a major catalyst and a validation of the drug's differentiated profile. With that, I'm happy to turn it over to Matt Lazarus.
Glad to be here and back in New Orleans. So we're making some forward-looking statements today. So PAS-004 is a next-generation macrocyclic MEK inhibitor. It's been designed to address the limitations and liabilities associated with earlier generation MEK inhibitors. If you're not familiar with macrocycles in general, they're a pretty big innovation in the small molecule development, and they typically exhibit superior target binding, better oral bioavailability, potency, and PK properties. There is a lot of opportunities to develop a MEK inhibitor. Pasithea is currently dose escalating in advanced cancer patients, and we expect to begin our study in NF1, neurofibromatosis type 1, next month. You know, the MAP kinase pathway is critical for multiple diseases, and currently approvals, there's been a recent tumor agnostic approvals for any tumor with a BRAF V600 mutation. There's opportunity in BRAF fusion, but there's also a lot of opportunity in rare diseases as well. Currently, we have a really good safety and tolerability profile, and we'll be rolling out more clinical data as the year goes on. We are now showing a greater than 60 hour half-life in human, and the two approved drugs for NF1 are both less than 8 hours. SpringWorks recently got a drug approved called mirdametinib. The brand name is Gomekli. That happened about 2 weeks ago. Their half-life is under 8 hours. And AstraZeneca is currently marketing Koselugo, or selumetinib, and their half-life is around 6 hours. And so bringing a very long half-life drug to this disease is going to offer, we believe, a constant suppression of the pathway, more dosing convenience, a once a day or less frequent dose, and you know, we're really excited about what we're seeing. And this is a really large market opportunity for MEK inhibitors. MEK inhibitors as a class are currently generating around $2.5 billion in sales, and the possibility for NF1, currently AstraZeneca is selling around $600 million for pediatric alone, and the adult opportunity is significantly larger than the pediatric opportunity. And 2 weeks ago, SpringWorks was approved for both adults and pediatric. So really excited about the market opportunity that we think we're bringing a best-in-class agent to. Our company is, the chairman is Larry Steinman. You guys may know Larry. He's the endowed chair of CNS at Stanford. He's one of the original founders of Neurocrine and Centocor. Tiago is a first-time CEO and a world-renowned researcher. And him and Larry were collaborating on another program, and is how they got to found the company. Dan Schneiderman, our CFO. And then recently we brought in Rebecca Brown. And we're really excited about Dr. Brown. She is a world leader in NF1. She recently became a professor at UAB, and you guys may know University of Alabama is a regional center of excellence for treating NF1, and she's going to be one of our clinical trial sites for our in the U.S. This is what our pipeline looks like. All of our focus at this time is going towards the MEK inhibitor, and so we're in dose escalation in cancer patients and soon to begin our NF1 trial. So quick mention about the MAP kinase pathway. I'm sure many of you are familiar with it, but it's essential for cell survival. And when it's abnormally activated, you can have, that's when the cancers or the rare diseases can occur. It's abnormally activated in many cancers such as melanoma, colon cancer, et cetera. And specifically, NF1 disease arises when there's a mutation in the NF1 gene, which encodes for neurofibromin. And so when that mutation is present, it basically releases the brakes, and so the tumors kind of grow uncontrollably. And all MEK inhibitors work the same. So when you are inhibiting or blocking MEK, you're stopping the phosphorylation of ERK. And this is a biomarker which can be measured. And so it's a really nice way to be able to confirm pharmacodynamic properties. Some of the MEK inhibitors are non-selective. So you may be familiar with a drug called trametinib. That drug hits MEK and RAF, and that brings additional toxicities. And I'm going to talk a little bit about the field in a moment. So NF1, there is still a very large unmet medical need. One of the really important features about NF1 is that patients really should be on drug for the rest of their life. And what we're seeing with the Zeneca drug, and we believe it's going to also be true for the SpringWorks drug, is that people drop off before 2 years. In fact, about half the patients are dropping their MEK inhibitor. And the reason for that is that 90% of the patients develop a rash. There's cardiotoxicities involved. There's a lot of diarrhea and nausea associated with the therapies. And so the ability to bring a drug that can titrate the pathway and deliver efficacy without having some of those toxicities, we think is going to be a game changer in the field. And NF1, even though it's a rare disease, it's still a more prevalent rare disease. There's about 100,000 people in the U.S. With NF1. And about 40% of them will develop what's called a plexiform neurofibroma. But importantly, all of them will develop a cutaneous neurofibroma. And I'm going to show you a picture of what that looks like. Prior to MEK inhibitors, the only way to treat the plexiform neurofibromas was through surgical resection. It's a tough surgery because the tumors derive from the nerve sheath, and they can cause pain and disfigurement, and they're wrapped around the nerves. And so the surgical resections are successful when they can be activated, but the MEK inhibitors have been a really big innovation to the field. And the cutaneous neurofibromas, this is what they look like. So on the right side is cutaneous neurofibromas. And importantly, we know when patients are on treating their plexiform neurofibroma, their cutaneous neurofibromas are shrinking. And currently, there's nothing approved for cutaneous neurofibromas. And this is a really big problem for the field. Cutaneous neurofibromas are disfiguring. They cause irritation, pain, discomfort. And then on the left-hand side, the plexiform neurofibromas, where there are the two approvals, they have a malignant transformation rate, in the order of 10 to 20%. And but for the most part, they're non-cancerous. And so it's important to be on a MEK inhibitor to keep these neurofibromas in check, because as they grow and they grow uncontrollably, they can touch other organs and cause all types of damage, pain, et cetera. Other manifestations of the disease just include seizures, headaches, renal artery stenosis, and GIST. So, how to improve on the existing treatment options. So I've told you already that the Zeneca drug, brand name Koselugo, has around a 44% response rate in pediatrics, and they are soon going to be filing for adult approval as well, but with a lower response rate. And then the SpringWorks drug, Gomekli, has around a 41% responder rate in adults and a little bit better in the pediatrics. The average tumor shrinks for the Zeneca drug around 28%. Both of them take around 7 to 8 months to generate a response. They're both dosed twice a day, and that's it can cause problem. And like I said, around 90% of the patients develop rash, and this does lead to a heavy discontinuation rate, where about half the patients are dropping off at the 2-year mark. So what are we trying to do with our MEK inhibitor? We want to hit the sweet spot between PK, tolerability, and potency. We've demonstrated a greater than 60-hour half-life in human. Macrocyclic molecules are more rigid with less off-target effect. We're seeing a really nice AE profile. We're going to believe we have a more easier compliance for dosing 1 time a day. And importantly, we're seeing a very low Cmax to Cmin ratio below 2, and that goes, shows a steady PK profile. The other MEK inhibitors have like a 10-to-1 Cmax to Cmin. This is a schematic which shows the kind of the evolution of MEK inhibitors. So all the way on the left, the selumetinib, that's a first-generation MEK inhibitor. That's the AstraZeneca drug. Then they made a bit of a modification to introduce mirdametinib. That's the SpringWorks drug. And then there was a company, Ardea, who was ultimately acquired. They had refamitinib. It's no longer being developed. And then our next-generation macrocycle. We're highly selective for MEK1/2. We have no primary alcohol. And in removing, this is really a big part of the technical innovation of our molecule. When we remove the primary alcohol, we see no active metabolites. All of the other MEK inhibitors have active metabolite, and active metabolites can confound PK and PD. And this is a schematic showing what I'm talking about here. On the left, you see short half-life drugs. So you've got significant fluctuation in plasma concentration. The target is not continuously inhibited because a 7-hour half-life drug will quickly lose half of its in the plasma. And the toxicities, we believe, are associated with the Cmax spike. On the right-hand side, this is what a long half-life drug would look like. You can hit steady state. We believe we can titrate the pathway. We have a very low Cmax to Cmin ratio, and we believe that's why we're seeing no rash, no ocular toxicities, and a much better profile. So currently, there are 5 approved MEK inhibitors. The top 2 are the only ones that are being used and approved for NF1. The bottom 3 are used in cancer. But it's interesting because the top 2 have failed, probably 20 to 30 cancer trials, where the bottom 3, are approved in cancer, but they're not tolerated in NF1. And so we're bringing an agent that's going to thread the needle. We believe there's also a broad application in combination therapy for cancer for our drug as well. And it's an active field. There are companies who are developing next-generation MEK inhibitors, but all of them have their own puts and takes. And only Pasithea's drug is selective for MEK1/2 with a very long half-life. And you can see some of the stark valuation discrepancies, and that's what creates the investment opportunity for Pasithea. Importantly, and we showed this in a mouse, when you have a dose-dependent inhibition of phospho-ERK, that correlates with clinical efficacy. So on the left, we're showing a mouse xenograft model with a dose-dependent reduction in tumor size, and on the right, you're seeing that correlates with a reduction in phospho-ERK. And we're going to be showing that hopefully in a human as well. This is another cell culture model where we compared head-to-head our drug versus the AstraZeneca drug. And what you see on the left is the AstraZeneca drug only inhibits the cells in NF1 models around 40%, whereas our drug gets up to a 90% inhibition while sparing of the wild type. And so we believe that indicates that we're going to have a better efficacy profile as well as a better safety profile. This is what our dose escalation looks like. And so we have currently dosed through cohort 4A and 4B, and we're now in cohort 5. And we're really excited about what we're seeing. We are advancing this in RAS, RAF, or NF1 mutated cancers. And it's a basket study. So we're getting a lot of late-line colon cancer, and also pancreatic and melanoma now that we've opened up some trial sites in Europe. So we've got 4 sites open in the US, 3 sites open in Europe, and importantly, we're getting some very interesting patients in the European sites. And so we'll have more to say on that soon. This is what our PK profile looks like. And I'm going to spend a little bit of time talking about our PK compared to the others. But as you can see here on the left side, as we've dose escalated, we've seen a linear PK rise, which is exactly what you want to see as you dose escalate. We have a low Cmax to Cmin ratio of below 2. It's actually about like 1.5 or so in this schematic here, and a half-life of greater than 60 hours. This is our PK profile as compared to the AstraZeneca drug. And so I'll just start on the bottom. At steady state, you can see the AstraZeneca drug goes from around 100 up to over 1,000. And then in the green, that's their active metabolite. And so we believe the active metabolite is what's contributing to that 90% of the patients with rash and some of these non-tolerated AEs that's leading to that high discontinuation rate. Whereas on the top, you can see our drug at steady state, that's the day 22 steady state, we just look like a straight line. And so, as we've dose escalated, we believe we're going to be seeing a corresponding titration of phospho-ERK. And so we believe for NF1, you don't necessarily need 100% pathway inhibition, that somewhere in the 60 to 70% range will deliver results without AE. And so that's what we're talking about here. More compliance, less AE, better dosing. So this is an analysis of SpringWorks drug, Gomekli, or mirdametinib. And what you see is that area under the curve exposure correlates with treatment response. And so in that circled area, the little red circles indicate partial responses, and the blue triangles indicate no responses. And so you can see when they get to around 700 nanograms per milliliter, that's when they start to see all of their responses in NF1, the tumor shrinkage. And so you take the SpringWorks drug twice a day. So if you double that 700, call it around 1,400 nanograms per milliliter is really where they're seeing their responses at 0 to 24 hours. And what you see on the right is that our 15 milligram capsule is already delivering over 2,200 nanograms per milliliter. And our 4 milligram tablet, which is we believe is going to be our final commercial formulation, is already a 1,500 0 to 24 nanograms per milliliter. And so really exciting to be delivering these high rates of exposure without seeing the rash and the AE. Graphically, this is what we're talking about. And so on the left, this is the SpringWorks drug in green, and the Pasithea, PAS004, in blue. And so you would take our drug once a day, and it's just a straight line, as opposed to the SpringWorks drug, which you're required to take at time point 0 and time point 12. And you can see that very high Cmax to Cmin ratio, which we believe is contributing to those toxicities. And on the right-hand side, that's the AstraZeneca drug, selumetinib. Similar story. You take it twice a day, it's got high toxicity, it quickly falls out. Now, the SpringWorks drug is a lot more potent than the AstraZeneca drug, and our drug is, we believe, to be equipotent to the SpringWorks drug. And so it's, you have to take the Zeneca drug at 25 milligrams twice a day to get a pretty high exposure to generate those responses, but they have a very high tox rate, and a lot of the people are dropping off. SpringWorks has a little bit better efficacy, but they take their drug 3 weeks on, 1 week off. And the reason for that is that they have these toxicities that require them to have a dosing holiday. And so far, we're not seeing any of those toxicities in our dose escalation, and that's given us a lot of reason for excitement. This is a view of the SpringWorks drug, mirdametinib, in their phase 1 dose escalation for cancer. And you can see they started at 1 milligram once a day, and they went as high as 30 milligrams twice a day. Now we have the drug approved. We know what their on-label rash is, which is 90%. But even in their phase 1 dose escalation trial for cancer, at their lowest dose, 1 milligram, once a day, 50% of the patients had rash, lots of nausea, fatigue. And you can see at the 4 milligrams twice a day, which is their approved dose for NF1, they had 75% of the patients with rash, 100% of the patients with diarrhea, fatigue, et cetera. And so these are tough to tolerate drugs. And so bringing a real innovation like ours to the field is what's driving all the excitement. This is what our tolerability profile looks like, and we can just focus all the way on the 15 milligram capsule. I would just point out these are very late line cancer patients, and so grade 1s are normal to see when you're on past 4 or 5 lines of cancer. You've got a colon cancer or pancreatic cancer, you're going to have some diarrhea, et cetera. But we are seeing very low rates of AE, no rash, no ocular toxicities, and this is giving us a lot of reasons for excitement with our program. This is what our NF1 trial looks like. And so we're going to be opening up 3 sites, Australia, 2 sites in Australia, 1 site in South Korea, and then the University of Alabama. And so we're going to be dose escalating. And one of the reasons and things that have come as a result of our cancer trial is we're going to be able to start our NF1 trial at a higher dose. And so we're going to be starting our NF1 trial at a 4 milligram tablet, which is a nice dose that we believe is already delivering a therapeutic effect. And so we're going to be kicking off that NF1 trial next month. And we're really excited to be able to show some interesting response data as the year unfolds. And so, this is an exciting program that's going to be moving pretty rapidly through the clinic. This is what our timelines look like. And so we have started our first in-human trial in the advanced cancer patients. We'll be kicking off the adult NF1 study pretty soon. And the pediatrics will then follow thereafter. We have to establish a little more safety in adults with NF1 before we can begin the pediatric cohort. But this is what it looks like. And then we're expecting that if all things go well, and remember, this is a known mechanism, so it's not a novel biology. We have a next generation MEK inhibitor. We've shown it in hundreds of cell lines as well as xenograft models, et cetera. We know MEK inhibitors work in NF1, and so it's about delivering that exposure that can generate responses. And we've already shown you guys today that we believe we're generating the exposures that will generate responses. And so we're very excited about how this looks, and we think that this drug will get to the market and then get a very nice piece of a very large TAM. As I told you, we believe the NF1 TAM is around $3 billion, $2 billion for plexiform and roughly $1 billion for the cutaneous. And the interesting part about the cutaneous and why rash is so important, well, you can't treat a skin disease by causing a rash, right? So the idea that we can bring a systemic MEK inhibitor to cutaneous neurofibroma when we know the mechanism works because we've already observed people who are on drug for their plexiform, their cutaneous neurofibroma shrinks. And so we view this to be a de-risked clinical pathway. There is a big opportunity for MEK inhibitors outside of NF1 and cancer. Within the bucket we call rasopathies, these are mutations in the MAP kinase pathway. There's NF1 plexiform, NF1 cutaneous. There's a rare disease called Noonan syndrome, where already MEK inhibitor is used off-label to treat these patients who have basically heart failure. We've done some interesting work looking at another rare disease called lamin cardiomyopathy, and we've shown dose-dependent reductions in heart fibrosis as well as survival benefits in lamin cardiomyopathy. Tuberous sclerosis is another active area, potential use of MEK inhibitors. The SpringWorks drug had a very successful study in mice. However, the short half-life drug in human isn't likely to stop breakthrough seizure, whereas a constant suppression of the pathway would likely stop the breakthrough seizure. And so we're considering some of these other rare diseases. And then in the case of cancer, we talked about the agnostic approval for V600 mutations, but there's opportunities in melanoma, colorectal, pancreatic, BRAF fusion, AML with RAS mutations. And so we're talking to a lot of different investigators who are interested in trying the drug and some of these other cancer indications. And then lastly, we're interested in what we call ETS2 diseases. And ETS2 diseases include IBD, ulcerative colitis, Crohn's, PSC. There was a really interesting publication in June of 24, so not even a year ago, in Nature. And like every good Nature paper, I mean, the data in it is overwhelming. But what they did was they ran a GWAS where they looked at over 4,000 molecules who would modulate ETS2 genes the most. And they saw that MEK inhibitors modulated ETS2 more than JAK inhibitors. JAK inhibitors are currently approved and selling, tens of billions of dollars treating IBD. And we know IBD is an ETS2 pathway-related disease. And on the right-hand panel in this Nature paper, what they show is that the MEK inhibitor is able to modulate cytokines as much as Remicade or infliximab, which is also an approved drug for IBD. And so the ability, so you can see here that the MEK inhibitor is modulating TNF, IL-23, IL-1 beta, IL-12, et cetera, all these cytokines which are hyperactivated in these ETS2 diseases. And so we think there is a real opportunity for someone to bring a MEK inhibitor to treat some of these rare diseases or even more prevalent diseases if you can titrate the pathway and dose them safely without some of these cardiotoxicities, rash, diarrhea, vomiting, et cetera, and ocular toxicities. And so we believe there really is a broad opportunity. And we're working with the author of this paper. His name is James Lee. He's out of the Crick Institute, to show that our drug also modulates ETS2. And so more on that coming soon. And so we do believe that there is had people, JAK inhibitors have such a wide use, and it's possible that MEK inhibitors, similar. Our intellectual property is really strong. We've got composition of matter on our original molecule with extension till 2037. But when we were manufacturing the molecule for GMP, we found a stable crystal form. We always presumed the drug was amorphous, and then when we were making it for the GMP manufacturer, we found the stable crystal. We filed all new IP. We ran our 6-month and our 9-month chronic tox studies. Most companies don't do chronic tox till you're in phase 2. In our case, we took it on at a very early stage because it's a known class of drugs. We just wanted to differentiate on the safety side. And so we've already completed 9-month chronic tox in dogs, 6-month chronic tox in rat. And we did that using our stable crystal form. And so we filed all the new IP around that. And, we work with Jones Day, one of the top IP law firms in the country. And so we have IP till 2045. So a really long runway with our molecule. We've also received orphan exclusivity for treatment of NF1, which is a nice thing that gives a nice round of exclusivity. And then we've got some new patent filings that we've been working on. And that really concludes my presentation. So if anyone has questions, I'd be happy to address them.